Alright, welcome to section 16.3. We're going to be talking a little bit about proofreading and repairing the DNA molecule. As we were covering in the previous section, the process of replication, it's important to remember that this is going on both sides. We have two templates and they're anti-parallel to one another. So if you remember, for the one template, we're going 5' to 3', which is the direction that DNA polymerase works in. You can only add DNA nucleotides to the 3' end. You cannot add it to the 5' end. So on the opposite strand, the lagging strand, we're using Okazaki fragments. Then as this builds, as we add these segments of RNA primers and then we build from the 3' end of the RNA primer, once we reach the next section, then DNA polymerase will start to replace these RNA primers with DNA nucleotides, and then ligase will fuse it together. But here's the question. Once this thing gets moving and we finally reach the end of the DNA molecule, okay, so we're going in this direction, and the helase just keeps opening up this DNA molecule, and eventually we're going to get to the end of it. Well, when we get to the end of it, how are we going to get rid of this RNA primer? Because there won't be room for us to attach DNA polymerase and take out these RNA nucleotides. That's going to be a real issue. And so before we get to that, we also need to talk a little bit about different mistakes that can happen. It's important to remember that DNA can be damaged in a number of different ways. There are a number of different chemicals. Cigarette smoke obviously is a big one, especially with lung tissue, but X-rays, ultraviolet radiation, there's a number of different chemical and physical agents and radioactive agents that can cause damage to the DNA molecule. And when this happens, there obviously needs to be some way for the nucleus of the cell to fix that. If it isn't, then you have a mutation, and that potentially could lead to cancer, could lead to all kinds of problems. So there is yet another enzyme that we're going to talk about in this chapter called nuclease. Its job is to cut out and replace sections of nucleotides that have been damaged. So as this nuclease molecule moves along the DNA strand, it is examining it, proofreading it, and when it comes across little bumps, little bubbles like you see here, then it knows something's wrong. It could be a bump because, again, remember, we're putting a purine with a pyrimidine, and that's what keeps the two strands at equidistance apart from one another. If we accidentally substitute a T for a G, now all of a sudden we have two nucleotides that are not going to match, and it's going to produce a bump, or it's going to produce a depression in that strand, and the nuclease enzyme is going to pick up on that. Its job then is to cut out that bump or that section that is damaged or mutated, whatever, and then DNA polymerase comes along and repairs the damage by attaching the correct nucleotide sequences that should be at these locations, matching the complementary bases with the other side of the strand. Now, you've got to keep in mind this DNA molecule, we're talking 46 chromosomes, it's gigantic. We're constantly replicating it as cells are growing and dividing, and so we're talking about a lot of proofreading that's going on. And to say that we're going to have an error rate of 0 is not realistic at all. The body does an incredible job of keeping the repair rate low, but it's not going to be 0. There are going to be sequence changes that could become permanent. Maybe it won't be permanent, but if it is something that's passed on to the next generation, then now we're talking about mutations that are going to potentially be a cause for evolution. Natural selection, I should say, which is the main driving force of evolution. If this mutation gives the next generation an advantage in the environment, over others in the population, then that's definitely going to favor that group of individuals that have the mutation to be successful, to breed and pass on their genes. All right, so now when we come back to what we're talking about with the replication getting to the very end of the DNA molecule, again, because we can't add to the 5' end, how are we going to get rid of that RNA primase that is, left as part of that lagging strand at the very end of the DNA molecule? This becomes a real issue for eukaryotic cells, not prokaryotic cells, because remember, prokaryotic cells, they have a loop of DNA, so you're always going to be able to come back to that 3' end. But with linear DNA, once you get to the end with the lagging strand, there is no way for us to come in and replace that RNA primer. Over time, as replication happens again and again, repeated rounds of replication are going to produce shorter DNA molecules, and the ends are going to be uneven. This is really going to be a problem over time. And so, like we were talking about, this RNA primer at the very end of the DNA molecule, removal of that primer, there's no way to add in another section of nucleotides because you can't add to the 5' end. So the second round of replication, this part is going to be left out, and now your DNA molecule is shorter. And so again, you go through the process of replicating, and again, you have a chunk that gets left behind. And with every successive daughter molecule that comes about through DNA replication, you are getting shorter and shorter and shorter DNA strands. You have special nucleotide sequences at the end that's called a telomere. Telomeres do not prevent the shortening of DNA molecules, but they can postpone the erosion of genes near the ends of DNA molecules. It has been proposed that when the telomeres get damaged or destroyed, again it could be through chemical or physical means, the shortening of these telomeres, that is one of the primary causes of aging. If chromosomes of germ cells become shorter in every cell cycle, if this just keeps happening, we keep leaving a fragment at the very end that doesn't get duplicated, then essentially we're going to start producing gametes where essential genes are going to be missing from the end of that strand of DNA. The enzyme telomerase catalyzes the lengthening of telomeres in germ cells. Again, the job of a telomere is to protect that end, to try and create a roadblock for this damage to get to the genes that are on the end of the DNA molecule. So there is an enzyme that will lengthen the telomere over time, and again, the purpose of that is to protect those genes towards the end of the DNA molecule. But again, if these telomeres become damaged, if they start to shorten, then now all of a sudden you run a serious risk of having cancer as an end result of cell divisions, because there is definite evidence of telomerase activity in cancer cells, and this is one of the primary reasons why cancer doesn't go through apoptosis. Cancer cells don't go through apoptosis. There's something wrong in that whole process. Finally, the last little segment here to talk about in chapter 16 is how the DNA molecule gets coiled up. Now remember, again, in a bacterial cell, we are talking about a circular loop, a plasmid, a significantly smaller piece of DNA than what we would have if we have this gigantic linear molecule that we find, and it is a double helix. I'm not going to try and draw that on the screen, but eukaryotic chromosomes are linear once they've been stretched out through the process of replication. Well, again, the cell, this thing is probably 6 feet long once it's completely unwound and stretched out. So you can't have 6 feet of DNA within a tiny, tiny nucleus. Therefore, we're going to have to get this thing coiled up, and so that's what the last section of the chapter is looking at, is how the DNA molecule gets put in a very supercoiled position so that it can fit into that very small space. Within bacterial cells, the region of the cell that the plasmid is hanging out is called the nucleoid. And the nucleoid then, once it becomes stretched out, is referred to as chromatin. Now, chromatin is different in bacterial cells compared to eukaryotic cells. In eukaryotic cells, there's a little bit of protein, but mostly it is just DNA. There's not a real need for a large amount of protein in their chromatin because the molecule is so small. But within a eukaryotic cell, we need to get this very, very large, long, fragile molecule bound up in such a way that it cannot become tangled within other stuff in the nucleus, other stuff in the cell as it is going through mitosis or meiosis. So we have these proteins called histones that we bind up into little balls referred to as nucleosomes, or beads, beads on a string. And it is these nucleosomes that we just wrap the DNA molecule around again and again. And we really, by tightly winding this molecule up and then taking those beads and winding them up, balling them up into loops, then we can really condense this very long, fragile molecule into a short, thick rod. And when you consider how much winding and coiling we are doing, it's no wonder that we can now see these chromosomes underneath the light microscope. We don't even need a high-powered piece of technology to view them anymore because they've been packed so tightly together. And again, this packing is happening during interphase. Now, some chromatin is organized into 10 nanometer fibers, but most of your eukaryotic cells are going to take that fiber and they're going to loop it and compact it even more, fold it and loop it into 30 nanometer fibers. And that's how they're going to create some space in the nucleus so it's not so jam-packed in there. These highly condensed regions are considered to be a restricted region in the nucleus, so as things are moving in and out of the nucleus, they're staying away from that area so as not to disturb the chromatin. Alright, so lastly here, most chromatin is going to be loosely packed in the nucleus during interphase, and then as we approach the beginning of prophase, it's going to start to condense and become a much highly condensed version called a chromosome. And again, a chromosome is made up of two chromatids, so chromatin, as it becomes bound up, turns into chromatids. The dense packing of this heterochromatin makes it difficult for the cell to express genetic information at this time. So in other words, we're not going to be building a lot of proteins as we enter into the early stages of prophase. Some of these histones can undergo chemical modifications that would result in changes in how the chromatome is organized. We're talking about here.
Several levels of DNA packing make up the compact chromosome seen in a metaphase cell. Even when a cell is not dividing, some of the DNA is folded tightly. The degree of DNA packing makes certain genes available or unavailable for transcription. Chromosomes consist of loops of supercoils, which are made up of tight helical fibers. These fibers are composed of nucleosomes, which consist of DNA wrapped around protein molecules. In the fiber, the DNA is unavailable for transcription. Each nucleosome is a cluster of proteins called histones. Nucleosomes change shape and position to allow RNA polymerases to move along the DNA and read the genes. Chemical modification of DNA and histones may affect chromatin structure and access to specific genes.